हेलो फ्रेंड्स, वेलकम बैक टू माय यूट्यूब चैनल। इस वीडियो में हम एमएससी जूलॉजी फोर्थ सेमेस्टर सब्जेक्ट बायोकेमिस्ट्री का टॉपिक यूटिलाइजेशन ऑफ क्रेब्स साइकिल का स्टडी करेंगे, जिसका इंट्रोडक्शन है। द क्रेब्स साइकिल आल्सो नोन एज द सिट्रिक एसिड साइकिल्स और द ट्राई कार्बोक्सिलिक एसिड TCA cycle is the fundamental metabolic pathway that play a central role in the biochemical processes of living organism. It is primarily involved in the oxidations of carbohydrates, fats, and proteins to produce energy in the form of ATP, adenosine triphosphate. This cycle occurs in the mitochondria of eukaryotic cells, and it is considered one of the key metabolic pathways that connect several biochemical reactions, contributing to cellular respiration. The Krebs cycle was discovered by Sir Hans Adolf Krebs in 1937, and since then it has been recognized as a crucial part of cellular metabolism. The cycle is not only vital for energy production but also provides intermediates that are essential for biosynthetic processes, making it a hub for various metabolic pathways. This essay will delve into definition, component steps, significance, and overall utilizations of Krebs cycle in biochemistry, especially for the perspective of zoology. First, definition of Krebs cycle. The Krebs cycle is a series of enzyme-catalyzed chemical reactions that occurs in the mitochondria of eukaryotic cells, where organic molecules are oxidized to produce carbon dioxide, water, and energy in the form of high-energy electrons carriers NADH and FADH2. The cycle is named after Sir Hans Krebs, who first elucidated its function in cellular respiration. The cycle begins with acetyl coenzyme, a molecule derived from carbohydrates, fatty acids, or proteins, which combines with oxaloacetate to form citrate. Second, the step of the Krebs cycle. The Krebs cycle consists of 8 distinct enzymatic steps. First, formation of citrate. Acetyl coenzyme combines with oxaloacetate to form citrate through the enzyme citrate synthase. Second, isomerization to isocitrate. Citrate undergoes isomerization to the form isocitrate through aconitase. Third, oxidative decarboxylations of isocitrate. Isocitrate is oxidized and decarboxylated by isocitrate dehydrogenase to form alpha ketoglutarate, releasing CO2 and reducing NAD plus to NADH. Fourth, oxidative decarboxylations of alpha ketoglutarate. Alpha ketoglutarate is further oxidized and decarboxylated by alpha ketoglutarate dehydrogenase to form succinyl coenzyme, generating NADH and CO2. Fifth, conversions of succinyl coenzyme to succinate. Succinyl coenzyme is converted to succinate by succinyl coenzyme synthesis, producing GTP, which is readily converted to ATP. Oxidations of succinate to fumarate. Succinate is oxidized to fumarate by succinate dehydrogenase, generating FADH2. Seventh, hydration of fumarate to malate. Fumarate is hydrated to form malate through the enzyme fumarase. Eighth, oxidations of malate to oxaloacetate. Malate is oxidized to regenerate oxaloacetate by malate dehydrogenase, producing NADH in the process. The cycle then repeats utilization with new acetyl coenzyme molecules. Third, energy productions in the Krebs cycle. The main goal of the Krebs cycle is the production of energy-rich molecules that can be used to generate ATP, the primary energy currency of the cells. The cycle itself does not directly produce ATP, however, it produces NADH, FADH2, and GTP, all of which play a vital role in the electron transport chain, where ATP is ultimately generated through oxidative phosphorylation. For each turn of the cycles, 3 NADH molecules are produced, 1 FADH molecule are produced, 1 GTP molecule is produced, which can be converted into ATP. Thus, the Krebs cycle plays a crucial role in cellular respiration by linking the oxidation of substrate such as glucose, fatty acid, and amino acids with the generation of highly energy electron carriers that fuel the electron transport chain. The role of acetyl coenzyme in the Krebs cycle. Acetyl coenzyme serve as an entry molecule into the Krebs cycle. It is derived from several metabolic sources including carbohydrates via glycolysis, fatty acids via beta oxidations, and proteins via deaminization. Once produced, acetyl coenzyme combines with oxaloacetate to form citrate, which is the first step in the cycle. Therefore, acetyl coenzyme is central to utilization of various macromolecules for energy production in the cells. Metabolic interconnections. The Krebs cycle is not isolated, it is tightly interconnected with other metabolic pathways. It links carbohydrate, fats, and protein metabolism through the shared use of acetyl coenzyme and intermediates such as oxaloacetate. For instance, carbohydrate metabolism, glucose is broken down into pyruvate in glycolysis, which is then converted into acetyl coenzyme and enters the Krebs cycle. Fat metabolism. Fatty acids are broken down into acetyl coenzyme through beta oxidations and enters the Krebs cycle. Protein metabolism. Amino acids are deaminization and converted into intermediates like alpha ketoglutarate or succinyl coenzyme, feeding into Krebs cycle. These interconnections make the Krebs cycle a central hub of metabolism, providing energy and intermediates for a wide range of cellular processes. 6. Regulation of Krebs cycle. The Krebs cycle is tightly regulated to ensure efficient energy productions. Key enzymes such as citrate synthase, isocitrate dehydrogenase, and alpha ketoglutarate dehydrogenase are regulated by various factors. Allosteric regulation. For example, NADH and ATP act as inhibitors of isocitrate dehydrogenase while ADP activates it. Product inhibitions. High concentrations of cycle products NADH and FADH2 inhibits the enzyme of the cycles, preventing overproduction of energy carriers. Feeding mechanism. The availability of acetyl coenzyme and oxaloacetate also affects the cycle activity. 7. Clinical relevance of the Krebs cycle. The proper functioning of the Krebs cycle is essential for cellular health and survival. Disruptions to the cell cycle can lead to metabolic disorders and disease. For example, deficiencies in key enzymes can impair ATP production, leading to conditions such as Leigh syndrome, a rare neurological disorders. Additionally, certain cancer cells show altered metabolic pathway, including change in the Krebs cycle as part of the ability to proliferate rapidly. Importance in zoology. In the field of zoology, understanding the Krebs cycle critical for comprehending how animals generate energy, grow, and maintain cellular functions. The cycle is not only present in human but also found in a variety of animal species. From invertebrate to vertebrate, Knowledge of the Krebs cycle helps explain physiological processes like muscle contraction, digestion, and organ functions, as they all rely on energy produced by this cycle. Conclusions. The Krebs cycle is a vital metabolic pathway that contributes to cellular respiration by producing energy-rich molecules like NADH and FADH2, which are crucial for ATP synthesis. It is a central hub in metabolism, connections the breakdown of the carbohydrates, fat, and protein of the production of energy. The cycle is tightly regulated to ensure efficient energy production and is essential for normal cellular and organismal functions. As such, it is of great significance in the zoology of biochemistry, especially in the context of zoology references.